uncharacteristically quiet? Thank you.
So I think it was in Garrett's talk that he showed the slide about how one of the great threats in drug approval is the failure to transition from phase 2 to phase 3. And then we moved, and then later on the same slide, I think he said that the problem is that we've reinvented phase 2 trials as pseudo-efficacy trials. And then we framed that problem as almost as if it's one of a technical information problem, that maybe we don't know how to design the right kind of phase 2 trial, but it might be that we're perfectly capable of designing the right phase 2 trial. There are other pressures that stop us doing it. And so one theme is this issue of instead of thinking we can educate ourselves out of the problem by giving us more knowledge about how to design better phase 2 trials, maybe we should actually look at why are we doing pseudo-efficacy trials, and list, going back to sort of the Levitt-Diamond issue, what are the people factors that make us repeatedly do pseudo-efficacy trials? Study sections that only want to approve studies that suggest they're going to improve outcomes, journals that don't want to publish trials that are changing sort of non-patient-centered outcomes, or even patients that no one wants to consent to be exposed to a risky drug when apparently the study has no meaningful chance of finding sort of improved patient outcomes. So instead of talking about creating positive enablers for doing the correct trials, also make a list of the negative disablers that stop us doing them. Because if we only make a list of how to do better trials, we'll still find ourselves in 20 years knowing how to do better trials, but not actually doing them.
I think that's an important point, and the time and again I see people doing phase 2 trials that would require 1,000 people in both groups to have any chance of showing efficacy, and they're putting 25 in each group in. And the reason for that is that frequently, particularly in small companies, they need to raise their next tranche of money, and they want to have some sort of positive press release to do that. And of course, it's lethal, because when the press release comes out and says that it didn't work out, then they're often dead. It would be better, I think, if we defined phase 2 trials as an opportunity to examine the mechanism of the drug's action, but also to define the effect size that would allow a better a designed phase 3 trial, because you've at least got some estimation of the effect size. The attraction for small drug companies to persuade them to do that is they're a lot less likely to fail in that than they are in their current model, which is that they do a study and apply a p-value, and guess what? It's not positive.
I wanted to go back to your list of 3 observations, Alistair, and I was really loving it until you got to the last one, and but really I would suggest that that's reframed, because I think I absolutely agree with you that biomarkers aren't the catch-all solution for everything in drug development and in medicine. That said, I see the issue as really one of a lack of agreement on the evidentiary standards, that we tout one biomarker as a surrogate endpoint and another biomarker as having regulatory impactful, being a regulatory impactful biomarker for a label, and then another biomarker being exploratory and hypothesis generation. I think for something like HDL, there's few people who would have really wanted to approve an agent based on HDL raising alone. So I think it's sort of a it's really not an argument that anyone would have. But the issue really is, what are the evidentiary standards, and how can we agree in a multi-stakeholder environment to get there and say, yeah, this is a biomarker like blood pressure or hemoglobin A1C that we're really willing to put our money down on, and this biomarker like HDL has some issues, and we're not really willing to put our money down on it.
The problem is we don't know it has issues until we discover it has issues. HDL looked like at least as good a biomarker as almost any I can think of. We had very large studies showing that the higher your HDL was, the better off you were. in contrast to almost any one of the what I'll call investigational biomarkers that are out there right now. So we only discovered that there was a problem once the drugs that were used to increase biomarkers went wrong.
Right, but that's always the case. We even for hard clinical endpoints.
That's why we should. Well, it's right. Right. Well, it's more than that. I mean, it's more than that. We need to know two things. We need to know that you change the biomarker in the direction we'd like to change it, but we also need to know that the drug doesn't have some negative effect in addition to changing the positive biomarker, and we can only get that from a hard endpoint. I would argue.
The other dimension of that particular example, though, was that you had a drug that raised HDL and raised blood pressure. And because of the secular bias, people chose to proceed believing the HDL and figuring they could manage the blood pressure. And that's not quite the way things turned out.
With HDL, that doesn't appear to raise blood pressure to the same extent, at least according to the press releases.
Yeah, so building on that discussion and the previous comment, I mean, thinking back to the CAST trial, right? I mean, everybody wanted to develop the super specific sodium channel blocker. Well, that killed a lot of people. So it's a matter of understanding mechanistically your source of variability and how they are so with biomarkers, one particular biomarker, it's not a one size fits all, one biomarker telling you the whole story. It's how that sits in the context of the other biomarkers, the patient characteristics, the genetics, et cetera, the pathophysiology of the disease. So again, making a plug in for building the quantitative descriptions of those interactions of those sources of variability, and building on the previous comment, it'd be great to understand, you know, phase one, preclinical, phase one, phase two, phase three, not as individual buckets, but as a continuum. And the more we move in that direction, we might end up with more seamless type designs and more informative approaches in which we could potentially use a Bayesian approach to use each piece of information as a prior that informs the next step, and then feed it back and confirm if that actually was informative or not. So would you agree with that, or?
I would, and I don't want to come over as negative on biomarkers. I think biomarkers have could potentially hugely enrich the populations we're studying. For example, we could use biomarkers to identify high-risk individuals or low-risk individuals, for that matter, to enrich the population that we study in a clinical trial, and therefore decrease our sample size and increase the effect size that we see. So there are various strategies that we can use. My point was a very narrow one, that we need to be careful that we don't lose sight of the reason we give patients drugs. We give patients drugs to improve their health, and we need to demonstrate that we have actually improved an outcome that matters to patients by the administration of the drug, and that's different from biomarkers.
Were there any comments from the panel on this? If not, let's.
I think I'd like to
Martin first.
defer to Martin first.
And to finish your point off, Alistair, I think one not only wants to know that what the drug does to the efficacy outcome that you have in mind, but in particular what it does to all the safety outcomes that you probably don't have in mind. And I think it's Bob Temple's quote, isn't it, that there's no surrogate for safety. And so I think when the biomarker literature is confusing, HDL, blood pressure, what the hell's going on, the IVA studies and so on didn't help us much either, then I don't have a problem with people doing trials. What I have a problem with is people prescribing treatments when there are no trials. And I think that that's the flip side that one needs to be very careful of. And I would urge that when one thinks about the gold standard, and I'm not sure that it's necessarily the most valuable gold, that one I wouldn't dare call it fool's gold because of who put the standard up. But when one thinks about that, actually, one has to think about the flip side, that actually there are opportunity costs to not evaluating medicines, whether they're new medicines, which I recognise is the FDA's concern, or they're the myriad of old medicines, some used for different purposes, actually some used for the purpose that they keep being used for, and doing trials. But I wanted to come back to the, what are the steps and what are the key things that need tackling. I think what I've learned over the last day and a half is that it's critical to be able to pose the right question. And that level of thinking and insight about what is the question that this particular experiment is, or that we're trying to address, that's the first thing. And secondly, what is the experiment that might help us address that question? And regardless of what experiment design you come up with, what are the errors that go into that you might have? For modeling, it's sort of easy. You know that modeling is wrong. The question is only how wrong. But for many of these other areas, the issues are much less well stated. So I think the first thing is about posing the question, and actually then working out and understanding these new technologies. And so that brings me to my second point, which is that actually understanding these new technologies, and I'm going to stick, for example, with big data, and I don't mean just sort of snazzy big data, I also mean electronic healthcare records and mini-sentinel and social media and all that other stuff. There's a huge amount of work there. I think Rob Califf yesterday called it the data janitor role. He said that he was proud to have been a data janitor. But I think that there's a whole issue about why would people go into that role and what does their career path actually look like? There's a sort of assumption in the room that somebody else is going to do it. Having spent the last 10 years organising the team that put the UK Biobank Health Informatics programme together, I know that there are people under the hood who have donated the mid part or the early part of their career for a decade, and who, without their efforts, the sort of high-profile GWAS or others results that are now coming forth, seems to be one every other day, and that's fabulous. Those sort of data resources require those academics to have put in all that work. It's not clear to me that there's a clear academic reward strategy for them. They will not appear at the top or bottom of the publication. Their H-index or whatever metric is the flavor of the day will be poor. And so in a sense, the question is, why will those people go into it? Now, the people I've got, I'm very lucky with the people I've got. It's because they're passionate about the type of science. Passion's great, but there's got to be a sort of reward for that sort of passion. And so one thing I would urge the panel to think about is how could there be some metric, some change of approach that, whether it was with the journals or whether it was with the way that academia credits its academics, is to think about reward structures for retaining those sorts of people. And I think that that's equally true for regulatory science. We heard several speakers say, I got into it for luck. My academic supervisor said you must be mad. Well, why is that the case? Why is that any less academically valuable than the many other things that academics do? And you could list a lot of things that academics do that are probably not terribly valuable. So I think the question is, for regulatory science, is exactly the same. What are the academic reward structures that actually bring those people on? Industry can take them on. Regulators can take them on, but you're trying to attract people into this career at an early stage. And during that phase when they're in academia, they have to be able to seen to be progressing and rewarded, and that's a challenge. And I think my final point would be, if regulatory science wants to be considered as regulatory science, it has to do the scientific thing and evaluate itself. So this comes back to that gold standard. Actually, we need to be, in everything that we do, evaluating actually the current state of play, some comparison with some alternative, and actually using that with judgment, that's what science in a sense is about, to actually improve. So I think the third element is evaluation.
So before you disappear, Martin, you and others in the audience who have published in the sphere of regulatory science outside of the big splashy clinical trial, how easy is it to get a run-of-the-mill regulatory science paper into the big journals? Because I will tell you, as an academic administrator who relies on external peer review of my faculty for promotion and tenure, we're all looking, we're counting the Cell, Nature, Science, and New England Journal of Medicine papers. So how easy is it to get the high impact paper in regulatory science?
Almost impossible. There are the occasional exceptions where a particular a viewpoint article or a particular sort of seminar series might actually get published, but they're few and far between, and they tend to be very focused or a particularly already expert and well-rewarded group who are putting those things together. If you're an up-and-coming academic researcher in regulatory science, then your top-ranked journal has an impact factor, whatever that means, of somewhere down there, below geriatrics and below many other specialties. So I think it's a major issue.
But frequently above toxicology.
Yeah.
So,
I wonder if that's the right question, Martins in plural. Maybe if we thought about expanding the quality of the questions we were addressing by addressing more mechanistic issues earlier, we would attract people into defining the right questions that would be really interesting at an earlier stage, and that would be publishable. The second point, I don't know if Sam is still here. The second point was back to Martin's career development question. It would be interesting, but I think he's left to hear how Northrop managed to create career. Is he? Oh, yeah. So maybe he should respond to how you create career paths for data analytical people and so on, because obviously this is a need for industry in addition to. Sam?
So I thought Martin's points were right on in many ways, or should I say spot on. So I think you actually sort of had the answer in your question, which is people who are trained in these fields can go into industry afterwards. They can go into agencies afterwards. But what do you do during that interim period where they're getting their degrees and then moving into academia before they move off into, say, industry or other areas? So I don't have necessarily a solution to this, but as an idea that we need to start thinking less about silos of industry, academia, agencies, and do a lot more of not just partnerships, because certainly we're doing that in multiple ways, but opportunities to have co-validation, maybe co-support that allows industry, academia to partner together so that the incentives are in both areas. We sort of have this very bright line separation, and for different reasons, obviously. But there has to be a way, particularly in these fields that are interdisciplinary, such as data science, regulatory science, where there's an opportunity for people to essentially free flow and get positive inputs and incentives from both areas. It's a thought.
I must confess some lack of clarity in my own mind as to how I would build that department in my school, right? And have faculty, getting back to Mark's point, we haven't talked about who's teaching the folks. And do we need to think about a slightly different academic department for education? And maybe that's one of the things that we leave for further development after this forum.
So I have two comments. One on this issue of training, which I'm speaking as Russ, the Stanford person, and then I have a IOM Drug Forum co-chair comment. So in terms of training, I think regulatory science is first and foremost science. Science is discovery of new knowledge, kind of period. Then when you say, well, what is the regulatory part? It's science, discovery of new knowledge, relevant to regulatory decision making. And so we have a strong bias so far in our efforts to make sure that people get independently excellent training in science. They can do science, and then you teach them at the end of that period and then transitioning into the next phase. And this is just our theory. There are other ways to do this. You transition them into understanding how to recognize a scientific problem that has high regulatory potential impact. And so the special skill you want to add after they're good scientists is the skill to recognize problems that will have maximal regulatory impact, because you always want to choose the most impactful thing you can do. And so once you decide that it's the space of regulatory questions that you're interested in, you need to learn how to say, that's a good question, but it's not of high regulatory impact and relevance. That's a good question that is of high regulatory impact and relevance. And so we're just at the beginning stages of trying to create this cadre of people who know how to do that, learning from our colleagues who've already figured it out. So that was just Russ from Stanford. Russ from IOM Drug Forum would like to encourage people to do exactly what Martin just did. So I was so glad to have deferred to him. He gave us 3 priorities. There is a meeting this afternoon of the IOM Drug Forum that is going to be thinking about steps going forward. How can we use our limited resources at the Drug Forum as a convener, as a forum, to push the regulatory science agenda? And so I'd like to especially encourage comments from the panel and from the audience on your top 1 or 2 things where we should spend effort. Don't worry about whether we can do it or not. We'll figure out if we can't do it, and we won't do it. But we need to know what needs to be done. And so I just want to encourage people to think about the take-home items for the next period of time.
Yeah, my name is Bill Potter. I'm a senior advisor at the National Institute of Mental Health, who has been involved, at least for the last 20 years, in many of these issues. And I just want to echo both what Martin Landry said and Peter Honig about how can you focus the really important questions that you would say fall under this umbrella of regulatory science, if your goal is to advance regulatory science, and properly reward the unsung heroes and heroines who have been involved in these tasks, which, as you all know, who have been involved in this, take frequently 5 years at a minimum, probably 10 years to see fruition and recognition. And I think for those of us who work on this, one of the greatest challenges has been to see the various stakeholders, whether they be the advocacy groups, the FDA, the NIH, or industry, actually support those individuals who are actively engaged in an important piece of translational work, bringing it to fruition and all, allowing them the time to stay in those positions. And a lot of the work, and I won't name the people, but particularly, and this isn't to criticize the FDA, but to point out that some of the people who've devoted the most time and energy to this FDA I've worked with, at least in the area I know most about, have been doing so after hours. They are not actually funded and supported to do this, not as part of their real day jobs. So unless our systems provide, just as Dr. Landry said, some way of rewarding these people, and maybe through all these high-level representatives we have. I mean, maybe we should be thinking across NIH, FDA, industry, pharma, even generating rewards for people, highly publicizing them, using your influence to see that these unsung heroines get a big people, whether it's an opinion piece, an editorial in major journals and all. There are ways of making this a little more respectable, but leadership needs to do it. Okay, and I know these people have not been getting the rewards, just like Dr. Landry said.
So if I might use the co-chair prerogative in deferring to some future time a discussion on the definition of regulatory science. I think we can all agree to disagree or hold our own part of the elephant. Can I just take a pause in the questioning and ask the panel, in response to Russ's request, what are the top 1 or 2 things that you would exhort the forum to think about?
I'd like to respond on a very high conceptual level. We're sitting here in the National Academy of Sciences building and trying to build regulatory science, and the comment therefore is science is the key. If you come at it from a societal point of view as compared to a scientist point of view, you could argue that regulatory is as important as science itself, because we could be sitting in a panel of lawyers who are interested in questions such as what are the ethical concerns regarding this? How am I going to make a decision on implanted embryos? How am I going to be doing all these other issues which are which are complicated from their point of view? So I really think that if you look at it in the way that I tend to look at it, which is not necessarily correct, but in the long run from society, not from how you're going to get a job, how you're going to publish, but what does society need? Society needs help on both ends of that. It needs help for scientists to understand some of the regulatory issues, but it also requires desperately regulatory people, that is to say lawyers, government officials, other people like that, to get education in science. And I just therefore feel I have to reject the concept that it's science with some regulatory. It's regulatory science because it's both. Thank you.
Thank you. Any other thoughts from the panel on the top burning issues that the forum should take on? Alistair.
I would stick with the ones that I sort of articulated before. I think defining and broadening the pre-competitive space is the most important thing. If we can improve our knowledge about the way in which the molecules or devices that we're studying are acting, we're going to be better off. And doing that in a way that brings in and encourages more people to think about the problem can't be anything but good. The second thing I would say again is that I think broadening access to the data in lots of ways would help. And Jeff Drazen and I had a discussion outside earlier about this, but for example, it's kind of incredible that we must have studied hundreds of thousands of people in control arms of trials, and nowhere is that data available. And forget drug development for the moment. Medical students, authors of papers would love to know what the natural history is of all these diseases we've studied, and yet that data has died somewhere. So I think we tend to always get pushback because we're worried about looking at the active arm of the trial, but just the control arm would tell us so much that we don't know right now across lots of areas, not just drug development, but across medicine, across, think of trying to define the value of care, how much better off we'd be if we had access to the thousands of patients who've been studied in the control arm and we knew something about the natural history of diseases.
Just to disagree a little bit with Alistair's last comment, those data are now available through the system that Perry talked about as well. So that, there are now 10, 12 companies that put their data up on the web. There were data available. As Perry mentioned, I
Right,
currently.
But what we need to do is to start to move to consolidate that data.
It is,
and in a way that is assumed to occur at every time.
yeah. Again, those companies that have joined that that version, that's exactly what is there. The data for those 10, 12 companies are all available. You can request companies' data from multiple different companies, so all within the same.
CPATH people are here, but it's worth just debating how difficult it is right now to combine the data from different trials, even in the control arm, and even in the trials that they took for the Alzheimer's trials, and where it was a fairly defined group. As I think they told us yesterday, it's very difficult to do that if it's not done carefully. And so there's a real work product to be conceived there.
Sorry, just curating these data are an enormous amount of work, as you're alluding to. I didn't mean to imply it's easy, but the data are available.
Good. And we're back to the data janitors, right? And who's going to pay for them? Darren.
Yeah, I was conceptual, so now I'll be very concrete. So it was kind of with a smile and a wave of the hand said that, when I was hired into the FBI, I have no idea how that process worked, but it's amazingly arcane, difficult, and many good people are lost because they simply just get tired of waiting and get tired of the bureaucracy. That, you know, a clear statement from this group, which is quite influential, that has to stop. That there needs to be clear hiring processes to get the best people possible into the workforce is incredibly important. So I would encourage that. Secondly, I don't see any reason why somebody can't just define. We believe as IOM drug forum that this is the pre-competitive space, and figure out what you think exactly that is and put it out there. And, you know, companies can react to it however they want to react to it, but have it on record that we think this is what the pre-competitive space is. So those are my thoughts.
Thank you. It's interesting listening from my perspective that much of the discussion has been on developing the external pipeline. There are a number of people within the agency already who are diligently honing their skills without recognition and without reward, and thinking about how those people can actually have an intramural career path that is both rewarded and recognized and allowed to flourish and supported. The other, I think it's been a grace note in some of the discussions, is the pay issue. So with company, I don't want to pick on Sam, but Northrop Grumman can afford far more than the FDA can. And so you get wage compression with bright young people coming in at very high levels of pay relative to folks who have been in the agency, have a lot of institutional knowledge for a long time. And so issues of internal culture and avoiding resentment in the ranks then becomes a real management issue. So I don't think we've really thought through some of these issues as new people, newly educated people come in and collide with experienced people in the organization. Frank.
Yes, I think we have clearly established that the dialogue between the stakeholders is very critical to move this forward. But I also want to point out that of all the partners and stakeholders in this dialogue, academia, in my mind, plays a critical and very important role. And I think, to be very blunt in terms of funding questions, when we talk about academia, we need to think about funding. And even within FDA, and this is all public information, when we think of sponsoring regulatory science and how much money we spend on regulatory science, when we count horizontal and vertical, we also tend to sometimes want to count cross-diagonal, because the money actually is relatively little. And you can all look up those numbers, and some of you will be surprised. This is seed funding, right? And as a society, we have to think about it. For example, we have no means to develop projects that are successful coming out of a regulatory science effort, and we cannot translate them, we cannot implement them, because there is no resource for it, and we haven't any resource plan for it. So this is not fundraising, this is stimulating to thought, just to be clear.
Two thoughts. Thanks for mentioning funding for academia. Coming from a place that is relatively well funded, Stanford, we still, I think, are underfunded. I'd like to talk about a couple of issues for regulatory science that directly bear on funding. It seems to me that in the next 10 years, cell-based therapies coming from industry are going to be in the future, if I read the tea leaves right. I can't imagine how they're going to be regulated. I mean, it's hard enough to regulate a single molecule. To start regulating and get consistency for cell-based therapies is going to be a bear, and that is going to require a ton of science. And most of that science and figuring out how to do it is in academia at the moment. And we really need to start planning about how to move what is basically basic discovery now into robust systems that can regulate cell-based therapy. And I would say that's something that is on the horizon, that isn't well, it involves big data maybe, but isn't necessarily exactly big data, but is a huge problem for society that is coming along if we want to do it right. The second comment I'd like to make is about biomarkers. Personally, I have trouble with biomarkers that have no biologic plausibility, and that is mostly biomarkers that are based on 10,000 transcriptional responses or, GWAS studies. I'm a good buddy of Russ's. We might argue about this, but I think you are on softer ground when you're dealing with that. Even when you have incredible biologic plausibility, as we heard, lipids, glucose, you can get really messed up by thinking that you can shortcut it. So I just want to reinforce what Dr. Ostroff said. Really, in the end, if you want to be sure that you're helping people, you got to do your experiment in people most of the time.
Well, what I would say in response to that is that I certainly agree with you, and I think one of the things that I was trying to say is that we've heard a lot of really innovative ways to approach decision-making over the course of this meeting, and some of them have been, quite frankly, spectacular. And the question for me is, to what degree do we look at these methods as both supplementing and supporting the more traditional ways that we approach our decision making? And at what point can we think about using them to supplant those ways? And that gets to a lot of what I think is being discussed here, because it's not an easy thing to answer. And so, certainly one of my priorities is the science around making a decision that you can use something like a biomarker to supplant some of the more traditional ways that we approach decision making. Or is it simply a tool that both supplements and supports the information that comes from other ways?
Thank you. To the left.
Thank you. Ron Kroll from University of Pittsburgh, former Chief Medical Officer at GlaxoSmithKline. You asked us to elaborate 2 or 3 things that we thought might be important. I'll pick one. Having participated in this exercise for so many years, I, at this stage in my career, feel we fundamentally fail patients and healthcare professionals at the time that we achieve approval of a new medical product, because we fail to answer the question that patients and healthcare professionals need to know the answer to, which is what's going to happen to me. So where I would look to try to provide emphasis here in trying to improve regulatory science or pharmaceutical medicine or whatever you want to call it would be to encourage the kinds of innovation in trial design, in biomarkers, in whatever it takes to try to get to the better answers to those questions, and to remove barriers to the communication of the information that exists. Those are barriers in regulation and barriers in speech, and also to provide incentives for the changing the approach from group population effects to personal effects.
In the interest of also throwing out ideas for regulatory scientists to think about, I'd like to make a case for artificial intelligence, for recognizing that the future of medicine is going to be massively data intensive, even at the N of 1 level. We're not dealing with population health, but precision medicine for an individual is going to require, within 10 years, processing networks of permutations and combinatorial processes that we can't even dream of today. And then these are going to be medical devices. This is going to be software-based computational tools that are going to help doctors make decisions on for a given patient. This is IBM's Watson computer has been a sort of metaphor out there for many of us, for many people, thinking about, well, someday artificial intelligence, doctors think someday the computer's going to do all of the stuff that I do. Well, it's time to get more serious about that kind of thinking. Number one, Watson is starting to enter the medical field very slowly, and I think, and at the population analytics level. But they recognize, as I think most of us do who are at all interested in the history of our artificial intelligence, know that it has to be part of N of 1. And yet this is an area of science. I'd love to see the Institute of Medicine convene some groups on this, because I think, and the National Research Council did one study about 2008, I think, that related to this. Use of, I can't remember what it was called, computational technologies. And I think this is just a huge gap in knowledge. And as an investor, I hear pitches from entrepreneurs frequently where they come in and they say, I really want to do an analytics kind of a program on the data that my patient has. And they want to use the EHR data. And they say, well, we can't do that. That data is so inconsistent, so unreliable, and so messy and noisy. We can't make any sense out of that. So already that data is too complex in a sense for this kind of science to go on. But what I'm talking about for the future is a much more complex area. So I think someone needs to get going on this science so that when people come to the medical device folks, whatever group that is within FDA, to say, evaluate this device, this software-based device that I've got, they'll have some science to help them do that.
Speaking about devices, and that group is called Center for Devices and Radiological Health, I'd like to thank the organizers for actually including devices into program and for using this medical product development in the title. I also have a proposal to make and would like to hear your thoughts about that. Would it be conceivable that the Board on Health Science Policy considers changing the name of the forum to Forum on Medical Product Discovery, Development, and Translation? And alternatively, if that's not possible, is it possible for us to think about creating another forum on medical devices? Just a thought for the future development. I think there are some things that we can heavily leverage from the discussion today, but there are also some unique things in devices world that potentially deserve some more collective thinking.
Hi, Carolyn Wilson from Center for Biologics. I wanted to make one plug for a priority area that I haven't heard anybody clearly articulate, which is, and I think Dr. Shakar really nailed it on the head, which is this whole concept of the data scientist. This is a really critical need when we're talking about this new innovative science that is coming our way to the agency. And then the other comment I wanted to make, which is in relation to a comment that Martin Filbert made a few minutes ago about how do we evaluate the kind of science that's done in the regulatory science arena. And I think that, you know, when you talk about publications in Science and Nature and Cell, you're right, that's not the right metric. And I think that another thing that would be helpful is to think about what is the right impact to look at. It's not the traditional impact factor that's important, but it's really the public health impact. And I thought the example that was so clearly and well articulated this morning about the QT prolongation studies just is one of dozens of those types of regulatory science findings that have a huge public health impact that aren't well articulated to the public, to the stakeholders, and I think we have to do a better job of articulating that and figuring out how to understand and measure it as well. And I think that that's what brings people into regulatory science, is that understanding of that public health impact of the work they do.
Yeah, I suspect if H factor stood for health factor, you might flip the priority scale.
Yeah, I think we need to be really careful, I suppose, because so many of us in this room are academics. We tend to view the standards by which we judge academics as the only standard, and I don't think that's fair. There are lots of other standards, but equally, I don't think we should try to shoehorn people who are pursuing different lines of inquiry into academic standards. Doing a good job in the job you're in should result in rewards that are appropriate to that job. It doesn't mean you have to do a job that Martin or I or someone else would necessarily think was terrific at University of Michigan or whatever. And to imply that seems to me to be denigrating the activities that other people are doing. You go to work for a bank, presumably you're judged by whatever standards the bank have. You go to work for Boeing, you're judged by whatever standards Boeing have. There are obviously excellent engineers at Boeing, and there are excellent engineers in a school of engineering, but they're judged by different standards, and that's okay, I think.
But I do think a deeper look at this cuts across both FDA and other regulatory agencies in which there are very poor bibliometric analyses that allow an agency to understand the impact of their intramural science program. And I think by definition, if you're doing a regulatory science effort, that it probably ought not to appear in Nature and Science and so on, because it has an entirely different applied purpose. But we do the research, and again, here we live and inhabit a faith-based organization. We believe that that research has impact in improving public health, but we have no direct analytic to show that.
Well, I guess what I'm saying, Martin, is I don't think we need to use the analytics that you and I may be most comfortable
I'm not
with
suggesting
doing. The engineer who designs a plane for Boeing probably doesn't, I don't know that, but probably doesn't publish very much, but presumably Boeing think he did a good job if he built the 787 or whatever it is.
They do, but the ATSDR only has not ATSDR, the folks that attend the crashes and plane crashes, NTSB. They only have the NTSB reports, which has a very low impact factor.
What I will say is that this is something that I think intramurally within FDA that we have been trying to define metrics for the impact of our regulatory science activities, and it's not the most straightforward and easiest thing to do, but ultimately I think what we've considered to be one of the essential features of measuring the impact is how the results have been used to impact regulatory decision making. And for us, that's probably the most essential feature or the most essential, like I think Russ hit it on the head in terms of what is the impact of that particular project or question. And so, you can use various types of metrics such as the number of publications, et cetera. But for us, it's how we then apply that particular piece of information to regulatory decision making.
So rising to the challenge of coming up with specific recommendations, would it be helpful then to say that we need a new system for evaluating scientists within FDA in terms of their creativity within the spectrum that they work, not saying that they have to publish in the NEJM or whatever other journal, but rather they have to come up with creative findings within your own spectrum.
That then get applied.
Right, that then get applied, right.
So I just wonder if we're underestimating the degree to which the most innovative users of data will increasingly be patients. And I think this is going to be a huge game changer in this business, and it's going to drive the behavior not just of academics, but also of industry and also the regulatory agency itself. We're used to telling people what they can safely take, what works. I suspect that increasingly this is going to be a dialogue, and perhaps that's something for Russ to think about, the unanticipated and anticipated impacts of individual patient empowerment on regulatory science and its deployment. If you know your genome harbors a massive amplification of what you think is a driver, you're not going to be interested in participating in clinical trials. You're going to try and get your hand on a drug that targets that driver by fair means or foul, et cetera, et cetera. So I think people will use all sorts of individual information sets to pursue individual innovative approaches to disrupting what we currently think of as regulatory science.
Kyle Meyer, CDRH. Steve, you said many of the things I was going to say. Jack Welch at GE notably talked to his community in terms of saying it's not, he had a very simple equation to talk to his engineers, saying that the impact of their work would be measured by the quality of their developments times the adoption or acceptance of their products. And we inside CDRH are, talking a lot about upstream metrics. That's not all that we would look at in terms of adoption of the regulatory science tools we develop, but it is something that we're working to measure. And I would put out there that we are seeing in many, from funding organizations and publications, a real kind of a stick approach to pushing for data sharing and software sharing, but I think we can do more as a community to encourage, incentives for sharing as well, in terms of how do we count it when an investigator gives away data or gives away models in addition to the publications that they develop from that work, because we all appreciate that when they give away their software, they're giving away, the ability of their competitors to be right behind them, making use of what they've worked very hard to develop and document. And in order to not have them hold on to that and just crank out papers from that, we need to find a way of giving them credit for that that is above and beyond publications.
Thank you. Well, we've now reached the end of what has been, I think, a remarkably productive 1.5 days of discussion, and remarkably on time. And I think on your behalf, I'd like to thank all the speakers and panelists and members of the audience who participated in what I think has taken us to a point that many of us were somewhat pessimistic we would reach when we started this process, but I think has the potential to improve the drug development process, accelerate the drug development process, and ideally bring drugs to patients faster. And for that, I thank you all. Thank you.
And I just want to take 10 seconds to thank Martin, Alistair, and the organizing committee, and I especially want to thank the IOM staff, the drug forum staff, and Claiborne, Chris DeFeo, who's been in the back left, at least most of there he is, Rebecca English, Emily Busta, and Katie Howell. Thank you very much for your efforts, and as you all know, they've been busy, and without them, these meetings don't happen, so we're very grateful for their efforts. And this ends the formal portion of our regulatory science workshop. Thank you.